welcome everyone again to another episode of the ideaXme show that show the profiles of the humans behind the really big ideas that are shaping our world and inspiring the future and future creators, and for all those that like really great stories. I'm Ira Pastor, your exponential health ambassador along for this journey. So on today's show, we are going to jump back into the themes of viruses and the human virome. As we've discussed on previous shows, viruses are infectious agents that typically consist of nucleic acids and protein coats. They multiply within living cells of a specific host, and the term virome generally defines a collection of viruses that make up a specific viral community. And on recent shows, we've touched on some interesting topics in relation to both the beneficial aspect of certain viruses, in the case of the show we did on bacteriophage therapies, and also certain pathologic disease states where viruses may be playing much more of a prominent role than previously thought, where we had a nice discussion with Ruth Itzhaki at University of Manchester on the viral connections in Alzheimer's disease. Today, we are going to delve into the fascinating topic of viral co-infections. So co-infections are broadly defined as sort of the simultaneous infection by multiple pathogens, multiple pathogenic species. In virology, co-infection includes the simultaneous infection of a single cell by two or more viruses, and a common example being the co-infection of liver cells with both hepatitis B and hepatitis D viruses, which can lead to so-called superinfections. And co-infection, needless to say, is a particular interest in human health because pathogen species can interact. There can be net negatives in terms of synergistic effects, but at the same time, there can be positive effects when certain viruses may suppress the virulence or colonization of another virus. Today, we are joined by Dr. Jack Stapleton, who is professor of internal medicine, infectious diseases, and professor of microbiology and immunology at University of Iowa College of Medicine, and director of the Levitt Center there for viral pathogenesis. Dr. Stapleton's research interests focus on molecular biology, epidemiology, and effects of co-infection in viruses including hepatitis C, HIV, and a very fascinating virus known as human GB virus type C, also known as hepatitis G virus, which we'll get into a little bit later in the show. In addition, Dr. Stapleton has clinical studies underway related to a variety of antiviral therapies, focusing on HIV, Hep C, and GBV-C. All that being said, Dr. Stapleton, thanks for taking the time to join us today on the show.
My pleasure.
We typically start off by giving our guests the floor for a little bit, just to talk about themselves. If you can just give us a little bit of a background on sort of who you are, how you developed this interest in medicine and infectious diseases, and ultimately how you sort of sit at the epicenter of this really fascinating dynamic of viral co-infection.
Like much in life, is very serendipitous. I wanted to do medicine and be a doctor, and during medical school, I was drawn to internal medicine and thought when I started my training in North Carolina, at University of North Carolina in 1980, that I would be a cardiologist. And 3 of my first 4 months, I was in cardiology floors, either ICU or floors that were preparing people for cardiac catheterizations and so forth. And I kind of came to the conclusion that it was very repetitive. And while it's very important and a good thing, I wasn't as attracted to it. And in October of 1980, a young woman came in and I was admitted to my service, and she had an unusual pneumonia. And I'd read about it in med school but never seen it. And we kind of did all the right things and diagnosed her within 24 hours with pneumocystis crenii pneumonia, which was unusual back then. And when I signed, she got much better. And I went home and I told my wife, this infectious disease stuff's pretty cool. There's good research going on and a lot of different opportunities, and people come in very sick and you can fix them and you can cure them. And I also like the idea of doing hospital-based service. And I said, and it's all in the hospital. It's all consultation and very little outpatient clinic. And I signed her diagnosis as it was what's called a myelodysplastic syndrome.
Mm-hmm.
Cancer, had Hodgkin's disease at age 14 in the mid-70s. And she ultimately came back several times with bad infections and died the following spring of 81. And by that time, I was seeing my third patient, or in June of 81, I was seeing my third patient in the year with pneumocystis. The other two were gay men.
Mm.
In retrospect, she died of AIDS before AIDS existed.
Mm.
And my care of her led me to go into infectious diseases for all the wrong reasons, because it wasn't curable. It was largely outpatient, but by the time I'd finished my training and I realized that I enjoyed taking care of people with HIV and AIDS people. So I was hired at Iowa to set up the HIV/AIDS program here and clinically. And my laboratory research during my training was on hepatitis virus, focused on vaccine development. So when I came here, I continued working on hepatitis A vaccines. And when the vaccine became licensed in the early 90s, I realized that in academic medicine and research, your goal in life is to put yourself out of business. And I say because we had a good vaccine. And so I kind of had to broaden my research. So I started on this new virus, hepatitis C, and I started doing some supportive sorts of virology laboratory work with HIV for our HIV clinic.
Okay.
So over the next 5 years, I worked on HIV work, and then in '96, this new virus was discovered that was related structurally, genomically, many ways it's related to hepatitis C. It's in the tree, the phylogenetic tree of flaviviruses, which is the group of viruses that these are included in. This was the closest human relative to hepatitis C. So I and every other hep C lab in the world started working on this new virus and pulling samples out of our freezers to see what it did. And over the first 3 years of the hepatitis G or GBV-C discovery, there were a couple thousand papers published, all pretty much clinically, epidemiologically related. And in '99, it became very clear to everyone that this virus doesn't cause hepatitis. But at that time, I had a brilliant postdoc and became research scientist with me, who made a full-length infectious clone of the virus. And we kind of discovered why it doesn't cause hepatitis. And that's because it grows in lymphocytes and not in blood cells like HIV and not liver cells. And we probably would have gotten out of the business, out of this virus, except there were two studies, small studies, one from Germany, one from Japan, of HIV infected who were studied for this virus to see if it caused liver disease in these patients, and it didn't, but it appeared to prolong their survival. And since I ran the HIV program, I had samples in my freezers from about 600 HIV patients, and was able to go in and look. And to my somewhat great surprise, these studies were real. You were 3 times more likely to be alive 9 years after coming into our clinic if you had the virus than if you didn't. So we spent the last 20 to 25 years trying to figure out why that might be and to learn more about it. And we've learned a lot about it over the years. So I don't know how much you want me to go into that, or if you have specific questions you want me to pursue.
No, absolutely. I appreciate that overview, because I'm going to go into that in the follow-up question. Before I get into that, I just want to sort of get sort of your top line sort of perspective on, what we'll call the beneficial virome, because we hear so much about the microbiome in the press nowadays, but less so the virome. But here we have this sort of extremely abundant source of these entities that outnumber everything else here. A large, whether 8-9% of our DNA is made up of ancient virus components. And while we typically think of a lot of these bad guys, the HIVs and the Ebolas and the Zikas, what have you, there is this whole other domain in terms of, I just call the beneficial, or the nice viruses, the good viruses, whatever you want to call them. How is this area sort of looked at? I mean, how do you approach this? Because obviously all the bad things we see in the press, we don't hear as much about, all the good things that viruses potentially are not just doing for our bodies, but elsewhere in nature. Let's talk about just sort of the topic of the beneficial virome, if you would, for a few minutes, and sort of how folks like yourself that study pathogenesis also look at this domain?
Well, so there are a number of viruses that are extremely common in humans, as you know. In addition to the endogenous retroviruses that are become part of your chromosome, there are many DNA viruses that, you know, the average number of types of herpes viruses a human has is between 3 and 5 because of chickenpox, Epstein-Barr virus, cytomegalovirus, HHV6, herpes 1 or herpes simplex 1 or 2. So people don't think about this. They don't think that, oh, I have 3 to 5 herpes viruses in me for life, and I don't have, and I probably have seen and probably have some papillomavirus in me, many different strains, and I have JC or papovaviruses, JC or BK virus. They only see disease from these when the immune system is knocked down and they're able to replicate to high levels. So those are clearly having effects on your homeostasis, your cellular milieu, and how much that bleeds into the circulation is probably extremely variable and depends on how much replication is going, or what many of these latent viruses actually produce latent transcripts to regulate. They make messenger RNAs and sometimes proteins that will be floating around, but they don't replicate and cause disease. Do alter disease. So Skip Virgin showed in a mouse model that a persistent infection with a murine herpes virus protected you against listeria infection, or protected the mice against fatal listeria infection. And it did that by regulating cytokines so they had more interferon produced. But too much interferon is antagonistic or is inflammatory. So that's not a good thing from heart disease and this. So there's probably a balance of inflammation that's protective, but actually over time is harmful. And we found with GB virus is that it actually dampens inflammation, and that's the punchline to our 25 years of working on this virus, is that it dampens but does not shut off your immune responses. So that if you get a strong enough stimulus, you make an immune response to whatever is triggering it, but it's not as severe. So you don't have overabundant inflammation. And I guess what's different about GBV-C and hepatitis C is that these are not in your genome, they're not in the nucleus, they're cytoplasmic viruses, so their RNA replicates entirely outside of the nucleus.
Okay.
And they replicate to very high titer. So if you measure the amount of this GB virus in the blood, on average there's 50 to 500 million copies of virus per cc of blood. It's a huge amount of virus, and yet the immune system doesn't react to the virus, doesn't clear it, and it does in many people, but not in about 20% of people who get infected. They don't clear this infection. They have this high level viremia for decades. Those are the people that are protected against HIV, because the way HIV leads to disease is through chronic inflammation and chronic activation of the immune system, which leads to decline, early death of the CD4, the immune cells that are protective, and that's why HIV causes disease. It's really not, it doesn't infect the same cells as HIV that we know of. I mean, it's possible that could happen, but no one's shown it. But it dampens the global immune responses, and that's been shown to be helpful in other diseases. There's a Ebola paper showing that if you have Ebola infection and don't have this virus, your mortality is 80%. But if you have this virus, it's only 20%. And there's a paper this year from a Swiss bone marrow transplant group that this may not just apply to viruses, that if you have GBV-C infection and you get a stem cell transplant, your likelihood of getting immune graft versus host disease, which is the graft reacting against the host tissue, the person who gets the transplant's tissues, it's reduced. It's not significant. It's a small study, but it's supportive of this idea that it's blunting immune responses and therefore preventing immune-based disease and transmitters. So I think this virus has been around a long time.
Right.
The data suggests that Lucy had this virus.
Okay. Okay. So I was going to ask you, that was my follow-up question, because, listening to you talk about it, it has to be one of the sort of the nicest, friendliest viruses that there could be. It doesn't do much to you, but it potentially protects HIV, Ebola, who knows what else. How ancient is this? Is this something that, has been around? I mean, you mentioned Lucy, of course, but does this, in the concept of sort of this, this viral symbiosis that occurs throughout evolution, is that, what makes this one so, nice, let's say, and are there others that you're beginning to study, whether they're the same, families or other types that are also demonstrating this type of, really beneficial symbiosis with us as humans?
So the first question about the ancientness of this virus, there are several lines of evidence that it's an ancient virus. First, if you look globally, the virus segregates genetically into groups, like most viral infections. So if you look in Western Europe, you have what's called genotype 2.
Okay.
If you look at different regions of the world, you find that their specific genotypes are predominant in different regions of the world. And if you look at the distribution of those genotypes, they actually reflect the migration of humans out of Africa. And then if you look at indigenous populations in South America, for example, they have the Asian genotype. But if you look at the mixed populations in the same area, Brazil and Colombia are two places where this has been looked at. The indigenous populations have the Asian genotype. The general population has a mixture of the Western European, North American genotype and the Asian genotype. And so we think that these, the Asian people who crossed the Bering Strait had this virus, basically.
Okay.
And then you look at animal viruses that are related, the New World monkeys and Old World monkeys have extremely closely related genotypes, suggesting that the precursor to the split of the continents had GBV-C. And if you look at the genetic distance between the genotypes of humans, it's essentially the same as the genetic distance between mitochondrial DNA of different races of humans.
Okay.
And then if you look, it's globally distributed. Even in remote islands in the South Pacific, this virus exists. So it's been around, and it's only been able to do that because it doesn't harm people significantly, or if it does, not until late in life. And there is a very slight increased risk of non-Hodgkin's lymphoma if you're persistently infected with this virus, but the risk is like 1 in 7,000 versus 0.7 per 7,000, all small risk. And that's the only untoward effect we've seen of this virus. But there are probably circumstances where you want extremely robust inflammation to fight an infection that might maybe not induce much inflammation. And this could be harmful in that situation, but we haven't identified that yet. So that was the ancient and the global picture. And there, you asked something else, I've forgotten what.
Well, I was just asking about if there are other related viruses or not related that show sort of this really extremely beneficial purpose in us.
It's not been well documented. There are, hepatitis C, which on average takes 20 to 30 years to cause disease in a minority of people who are infected, doesn't cause much harmful effect in the short run in the vast majority of people. And in some people, over a course of 30 years, the majority of people don't have any liver disease. And it has some similar mechanisms to dampen immune responses, which is partly how it persists, we assume. And so understanding those could be helpful, and that's, we study that area as well. But there are very limited data on any beneficial effect. There are a few abstracts from meetings in HIV-infected people that in early HIV infection, before the CD4 count has fallen, there's some suggestion that CD4 count may fall less fast, and that's a marker of HIV disease progression. But once the CD4 count's low, what happens is HIV then doesn't control Hep C as well, and it becomes more pathogenic.
Okay.
So we actually have increased pathogenicity of HCV and as immune compromise happens. So that's hard to say. I think it's an understudied question as to what role and like CMV, HHV6 have in other disease states. And I think you mentioned your Alzheimer's. Were you talking about CMV in that regard?
Yeah, I believe so.
Yeah. So CMV is certainly being implicated in the pathogenesis of other diseases, but I'm not aware of any where they're beneficial.
Okay.
The microbiome is not as expansive as bacterial, for sure. And that's probably because you have to be a successful virus, you have to adapt to a host cell.
Sure.
You can persist in that's very specific, and you just can't replicate freely in the GI tract, for example, or in the lungs. And so there's not the diversity, there's a lot of diversity, but there's not the expansive diversity of bacterial microbiome. So in people who are looking at microbiomes to discover new viruses, they're not finding lots. There's a group found a rhabdovirus that seemed to be in asymptomatic people in Africa a few years ago. And that's a very interesting finding. But I think a lot of people think, oh, if we look with deep sequencing where we can just shotgun and find every virus in there, we're going to find a bunch of things we don't expect. And that's not happening as much as you would expect. And I think it has to do with the restriction to a specific host cell.
Interesting.
And so there will be viruses found, no question. But it's not going to be as extensive and as diverse as you see with bacteria.
Gotcha.
In my opinion, that's a speculation.
No. you would know best. Moving on to the sort of the area of clinical work. Obviously, you're involved in a lot of different antiviral clinical trials, but thinking more about this co-infectivity topic. As mentioned, we've talked about the use of viruses in different therapeutic areas, obviously, as carriers for gene therapies and bacteriophages and of course attenuated vaccines and all this. Is there a sort of what are the possibilities in terms of this knowledge of co-infectivity for therapeutics? I mean, is it something where you'll be looking at this inflammatory dynamic? Is there ever a case where you foresee, hey, a cure is infecting somebody with X virus to balance out Y virus? What are some of the possibilities here that you're talking about?
So yes, one fact to know when you think about this is that every day in the United States, somewhere between 600 and 1,000 units of GBV-C infected blood are transfused into humans. We do not screen the blood supply for this. About 2% of healthy US donors have GBV-C. And because of the risk to the blood supply of losing donors and the lack of evidence that this is going to alter or cause disease, this decision has been made not to screen blood. So about 25 years ago, I proposed, we had a lot of patients with, maybe 20 years ago, we had a lot of patients with drug-resistant HIV. And I proposed to infect multiply resistant HIV-infected people with transfused blood that was licensed for use as blood products. And the FDA was very nervous about this. And they told me they thought it was an interesting idea, but what I was wanting to do was what's called a phase 1 study, just to show the safety of it and see if there's any measurable effect.
Sure. I
think this is, again, my opinion, and they would probably deny it, but I think no one wanted to be the person who signed the line saying, yes, we approve infecting people. And so they came back and said, if you want this to be scientifically valid, you have to do it in a blinded fashion. If you select donors knowing that they're positive, you're picking a drug. But so you have to do a non-screened donor, look at the results, and get back to see who's infected. And scientifically, that definitely stronger, but it meant that you'd have to infect 500 to 1,000 people with unscreened blood to have enough cases to see anything.
Right.
And the cost for that was unrealistic. And the people at NIH who I was funded by and that I knew and know quite well said, don't ask us for money to infect people. So I think nobody wants to be the person who signs off on the test if something bad happens, like test or something like that. So that didn't happen. I have colleagues who have tried this in Brazil, thinking it would be a lot easier, and it turned out it wasn't a lot easier in Brazil either. The nearest we can get to this is a study Mike Bush's group did, and I contributed to this some, was in HIV-infected people who received blood transfusions in the 80s and 90s.
Okay.
And in those people who were not infected with GBV-C and became infected with GBV-C, the survival was improved similar to what we saw in our clinic. So that's, I think, as close as we can get to saying that there's a post hoc effect of this virus on HIV-infected people. But so that was one of the reasons we used in our grant application for why we wanted to study this, was it could be a therapeutic. Second was it'd be a great gene therapy vector. You've got 50 million copies of virus floating around that doesn't make people sick and blunts the immune response to itself. And so we still like that idea. The problem is like Hep C and many of the hepatitis viruses, this virus does not grow well in cell culture.
Yeah.
It's almost impossible to grow in cell culture. We're probably as good as anyone at doing this, and it's very inconsistent and low level and inefficient. And so the ability to really study and prepare and produce virus that would be useful to insert foreign genes is not feasible at this point. My former students are working on this at different places, and I keep in touch with them, and I wish them well. And there are people that from the gene therapy world who call me periodically, and we always include everybody, because it took Hep C almost 20 years to, well, it took Hep C about 15 years to get a tissue culture system that would be used. And that was with multiple labs, dozens of labs around the world that were large, highly well-funded, and some luck to get a system that Hep C would replicate reproducibly in. In a field that's very poorly studied, where there are maybe 4 or 5 labs in the world that work on it still, and they're not well-funded, it's very difficult to really generate the type of push that's needed to come up with a system for developing this. But as new systems come along, we try them.
Sure.
If we had a good way to grow this virus, we'd be doing just that. That's one of the most useful potentials for this virus that's out there.
Gotcha. Well, I have you. I want to shift over. You mentioned early on, you were around doing your work at the very beginning of the HIV epidemic. Here we are a few decades later. Could you give us sort of your top level view on sort of where we are in 2019? I mean, we hear obviously about vaccines. We see some of the bone marrow CCR5 transfusion cures and things of this nature, but then also, we hear about new strains and still problems in the developing world. Can you give us your top line on sort of HIV 2019 and where the whole area is going, what you're most excited about, what you're most concerned about?
So I tend to be a skeptical person, not optimist, but skeptical, highly skeptical. And my opinion on the vaccine development is that a lot of smart people have devoted a lot of effort, and by and large, it's not been highly successful. And I don't see new vaccines that are totally novel approaches that I think are probably going to be needed if we're going to get a vaccine against HIV that will be broadly useful. So I'm a bit of a pessimist about vaccines. The cure, I was very much a pessimist a few years ago, but I think the advent of gene editing and CRISPR and different approaches for gene editing have really made such huge strides that I hope that those can be safely applied to humans and target the virus. And so there might be a cure through that route. There are other, I mean, we have effective therapy, which is, I mean, if you walk into our clinic and you're in your 20s, expectancy is almost identical to that of a person who doesn't have HIV if you take your meds. And the meds are much easier to take and simpler. So we have effective therapy. Whether or not we can make that a lot better with different approaches, antibody approaches or gene therapy approaches, remains to be seen, I think. But from a cure standpoint, I think the most likely beneficial thing is going to be a gene editing approach, where you cut out critical sections of the HIV genome and it becomes basically an endogenous retrovirus that doesn't cause disease. And so that's kind of my take on it. Now, I am not a cure person. They may have other issues that I'm just glossing over, but I'm not as enthusiastic about cure except through that approach at this point in time. It's funny, and I probably shouldn't do this on tape, but I'm getting older and I don't care much about what people think of me. But some of our grants have the reviews have said things like, well, this is really interesting, but there's not been much practical come out of the work on GBV-C. And I think, well, we're learning about inflammation and biology of a frequent common human infection, not pathogen. And then I think how much more has been spent on the cure and practicals come out of that. But you can't say that to the study section, but I'd like to. So I'm getting old enough that I don't care, and I'm just going to say, I think none of us. It's very difficult to predict what's going to be a major practical outcome of basic research, and so discovery research. I think you should not focus all on one area, and you should kind of wild hairs you should look into. So something interesting might come out of it.
Absolutely.
With the current funding climate, it's very difficult to do that.
Yeah, I can imagine. One final question while I have you, just going back to sort of the beginning. You mentioned originally you were looking at cardiology, and then chance took over. Any specific influencers throughout your career that you might want to give a shout out to at this point, that if it wasn't for them, I'd be talking to Dr. Jack Stapleton, cardiologist right now, versus infectious disease specialist?
Probably the major person was a guy named Fred Sparling, who was chairman of micro at UNC, Madison at UNC. Great guy, great scientist, really a creative thinker, and he got me interested in infectious diseases, and was really encouraging and gave me a lot of good insight throughout my training. And I went to his retirement party a few years ago, and he's still was still doing great work when he retired. And then my lab mentor, Stan Lemon, really taught me a lot. He's a really smart guy who has been a major player in Hep C and positive strand RNA virology, and understanding replication and particle structure and that sort of thing. And he's a really good thinker and writer, and I think he helped me more in those ways than anyone else. So those two people are probably my main influences.
Outstanding. I appreciate that. Jack, it's been a pleasure and an honor having you take the time to share your knowledge with us, and really, as we stay on the show, moving the story forward. And it's really fascinating work that you've done and have been doing for the last several decades. I just want to really thank you for that. And for everybody that is watching, but also going to be listening on the various podcasts, we've been spending time with Dr. Jack Stapleton, Professor of Internal Medicine, Infectious Diseases, Professor of Microbiology, Immunology, University of Iowa College of Medicine, and Director of Levitt Center for Viral Pathogenesis. Truly an amazing set of research, amazing ideas and thinking, and really, once again, thank you for everything that you've been doing for everybody throughout your career. It's truly a great story.
Well, thanks for asking me to do this. I always like to talk about it. It's I think it's pretty cool and pretty interesting.
It definitely is. Definitely is. Good talking to you, Jack.